Okay, so we're going to talk about Unit 5, and this is our second set of notes, so 5, period 2. We're going to be talking about osmosis. So, some of the things that you're going to know by the end of these notes are the words hypertonic, hypotonic, isotonic, lysis, what the ideal situation for an animal cell is, and the ideal situation for a plant cell. So, as you recall from middle school science and possibly integrated, osmosis is the movement of water. It's not any other particle or molecule, it's just water that gets moved. And so, we're going to talk about a little vocab here. So, the first word is solvent. That's the liquid substance, such as water. It doesn't have to be water, but it's a liquid substance that does the dissolving. Okay. And water happens to be the most common solvent, so we call it the universal solvent. Our next vocab word is solute. This is the solid, and it can be an example of salt, and it's the thing that actually gets dissolved. And so the solvent dissolves the solute. And together they form what's called a solution, which is a combination of a solute and a solvent. So just remember, a solution is a mixture. It's not a compound or a molecule because they're not chemically bonded. We still have two distinct things. We have the solute and the solvent, and they just happen to be mixed really well together. And so it's kind of uniform, or it doesn't look. It looks like one thing. And you can separate the mixture, but it's not so easy as just like picking it apart. You'd have to evaporate the solvent out, and what is left behind is the solute. So we're going to talk about this title here. Tonicity drives osmosis. So what is tonicity? That's the solute concentration. So just to kind of give you a little background, if a solution has a high concentration, that means it has a lot of solute and very little solvent. So the thing to think about would be kind of like orange juice that's frozen in those containers. Those have a high concentration because they have a lot of solute and very little solvent. Vice versa, a solution that has a low concentration would have a little bit of solute and a whole lot of solvent. So you could think of like if you're making Kool-Aid and you put just a pinch of the powder in, that would be the solute, and then you fill the whole pitcher up with water, that would be the solvent. So that would be a low concentration and be kind of like watered down. Okay, so kind of now that we understand what solute and concentration is, we're going to talk about these different environments our cells can be in. Okay, so tonicity. This is solutions in which the cells, when a cell is placed in the solution, it can be placed in one of three solutions. So a cell can be placed in what's called a hypertonic solution, which means it has a high number of solutes. So this would be a very concentrated solution. Or a cell can be placed in a hypotonic solution, which it has a letter O, so it's lower number of solutes. So this would be that very dilute. So a hypertonic solution, this would be a concentrated. And then hypotonics would be a very dilute, or a lot of the solvent, whatever that may be. Or it could be an isotonic, which means that the cell equals the solution, meaning they have the same concentration. So this next phrase right here, this is very important to know. Put some stars by it. So water always flows from a hypotonic environment to a hypertonic environment. So it always goes from a hypo to a hyper. Why is that showing that? Okay, it always goes from low to high to even out the spacing between the particles. So remember, this is a very dilute, and these particles. So if I want to show the particles in a dilute, maybe they're pretty far apart, like that. Show them far apart. And if I want to show the concentration of particles in a hypertonic solution, they're very, very packed together. It's very concentrated. So the water, which is in the space between these particles, will move from one to the other to cause these green particles to separate out. So, what is a cell in a hypertonic solution? The solution it's in has a high concentration of solutes. It causes water to diffuse out of the cell, and the cell will shrink. A cell in a hypotonic solution, the solution has a lower concentration of solutes that causes the water to diffuse into the cell, which causes the cell to swell. A cell in an isotonic solution, the solute concentration is equal to that of the cell. So what that means, there's no net movement of the water. Water will still cross the membrane, but whatever goes into the cell, equal amounts will go out of the cell. So there's no net, like there's no more of one direction into or out. The same amount that goes in is the same amount that goes out. So even though the water is crossing, it's just crossing at kind of a constant rate, which is known as dynamic equilibrium. So it means that the cell's not going to get bigger or smaller. It's just water's moving in and out at just kind of an equal rate. Oops, I need to go that far. Let's go back one here. Okay, so here's our examples here, and we need to talk about which one, where's the water going? So let's kind of look at this first one, A. So A, if I look at A, there's definitely more of the green particles on the inside of the cell than there are on the outside of the cell. And so what's going to happen is it is more concentrated inside, so the water wants to kind of separate. It wants to be in equilibrium, so the water is going to go into. So we would call this solution a hypotonic solution. The solution right over in here on the outside of the cell is the hypotonic solution. And therefore the water will go into the cell, which causes the cell to swell up. So our second example here, if I look at the green dots here inside the cell, are kind of equal spaced to the green dots outside of the cell. So water will still there's still a movement of water, and I'm going to go quick back to A. There's still amount of water that actually will move out of the cell too. It's just a lot more will go in than out. And so therefore the net amount of water will be going into the cell. So in this case, there's still water going in and out of the cell. Whatever goes in is going to equal the amount coming out. Arrows are the same side. So this solution is called isotonic. And the cell is going to stay the same. Because it is in dynamic equilibrium. Which means that water is going in and out of the cell, but whatever goes into the cell is equal to that coming out of the cell. So the cell stays the same size. Now in this case, in letter C, you can see that the solution in the blue is much higher concentration than inside the cell, which is the orange area. So this is called a hypertonic solution. Which means that the water from inside the cell is going to move. There's going to be a net movement of water out of the cell. Yes, there will still be some water coming in, but it's just a very small amount. That the stuff coming out is going to be much greater. And it'll do that until it kind of reaches equilibrium. And what happens to the cell is the cell shrinks in size because the water is going out of it, so it'll just get smaller. So again, here's my hypotonic solution. I'm looking at this first one here, hypotonic solution. The water will go into the cell, and it causes the cell, and this top row, this is an animal cell. So if you look at this animal cell, having some technical difficulties here. So if I look at this animal cell, these are the ones in yellow here. So when water goes into the animal cell, it's going to actually, it can sometimes move in so much that it stretches the cell so much that it becomes what's called lysed, or it starts to split. Now in a plant cell, remember plant cells have this cell wall around them. And so that's it doesn't lyse like it does in an animal cell because of the cell wall giving it structure, but it becomes what is known as turgid. And for a plant cell, this is really good. This is their normal state. They want to have lots of water in them. And you'll notice that when plants don't have a lot of water in them, they start to become limp. So turgid, this is the normal for plants. This is good. Lysed is not normal for animals. So that is not good for animal cells. You don't want them to be lysed. They have too much water in it. Our next solution is an isotonic solution. So that means it has equal water in as it has out. And in the plant cell, the same, equal in and out. Now, this is normal. So this is good for an animal cell. They want to have this equal in and out. But for a plant cell, it's not enough water, and so it's actually called flaccid. So this is not normal. And this is where your plant itself starts to kind of wilt. Last type of solution, we have a hypertonic solution. This is when the net movement of water is out of the cell. Yes, it could still have water going into the cell, but it's a very small amount. Most of all the water is going out of the cell, and it causes the animal cell to become shriveled. And when we have net movement out of a plant cell, the plant cell becomes plasmolized, and neither of these are good for a plant, or it's not good for an animal cell. So both those are kind of bad conditions. So the two to remember, the two normal conditions for plants, it's turgid, and for animals, it's in that isotonic solution. Plants, it's a hypotonic solution. Those are both good for each cell. Osmosis is a form of what's known as passive transport, which means it doesn't require any energy to occur. It's a natural process between these different concentrations. Move from the hypotonic to the hypertonic solution, and there's no energy required, meaning there is no ATP, which is the energy that we use in our cells.